As you can see from the screen here, it shows list of samples that are collected for tests in the lab. We have blood sample, stool sample, urine sample, sputum sample, effusion fluids, urogenital samples, and biopsy. Start by blood sample. Blood sample can be collected in order to be used for biochemistry tests. It can be used for plasma glucose. It can also be used to investigate non-functional plasma enzymes. These are enzymes that are released from a tissue as a result of damage. For example, liver damage, myocardial infarction, and also blood can be used for renal function test, but it's not non-functional plasma enzymes that is targeted for renal function test. For renal function test is creatinine and urea. Hematological tests, also blood can be used in hematology lab, where it can be used for sickling test, ESR, which is erythrocyte sedimentation rate, solubility test, full blood count, and differential count. Blood can also be used at blood bank during ABO and RH grouping, compatibility test, which can include flash frozen plasma test, which is abbreviated as FFP, and close match test. Blood can also be used in serological tests. Example of a serological test is rapid diagnostic test, which is RDT test, test for malaria, test for RPLR, test for coronavirus, test for HIV and AIDS. We can also use blood for immunological tests during viral load and CD4 count. Blood can also be used in microbiology lab, where we do blood culture when sepsis is suspected, and also parasitology for malaria parasites. Stool sample is also another type of a sample that is collected. So a stool can be used in microbiology during stool culture, microscopy, and sensitivity. In parasitology, stool evaluation is started with microscopy, looking for adult worms, and microscopy, the eggs for parasites, cysts, and larvae. Stool can also be used at molecular level, where we look for a genome of an organism, e.g. gene expert test for tuberculosis bacilli bacteria. Urine sample is used for chemistries, which include the use of urine dipstick, such as urinalysis and pregnancy test. Urine can also be used in microbiology for urine microscopy, culture, and sensitivity. Urine microscopy, that's why we see epithelial cells, cellular casts, white blood cells, red blood cells. Sputum. Sputum is used for tuberculosis bacilli identification on the microscope. Also, at molecular level, we can use sputum for a genome of tuberculosis bacilli in a gene expert machine. Urogenital samples. Examples of urogenital samples, we have vaginal swab, and a cervical swab, and a cervical swab. So these are collected when UTIs and STDs are suspected. Then biopsy. These are sample organ from a living being, from a living tissue, extracted from an organ. For example, the breast, liver, or any organ. It is used for histopathological and cytopathological purposes.